So really, it's very exciting for those of us who have been working on the genome and the interpretation of the genome, that is, the decoding of the genome. But the good news is, of course, we now have many genomes, not only of humans, but of many, many different organisms, and pretty soon of many different humans, so that we can really understand comparative genomics. The bad news is that so far we've really only learned to read about 2% of the genome. So I'll give you a very crude analogy. So the genome may be not so much the book of life. I think of it more like the yellow pages. And we're trying to figure out how the telephone system works by reading the yellow pages. It's very difficult. And not only that, but we've only figured out how to read about 2% of the yellow pages. So there is a very big challenge. Some of you may have heard from Mike Bishop's comments about these very important genes and gene products, that's proteins, regulators, that can cause cancer. These are the things that accelerate growth or things that slow growth down. A lot of the same molecules that are involved in cancer are also involved in regulating gene control and gene expression of all genes. And so a major challenge that faces us as we move forward in trying to interpret genomes as a way to predict disease susceptibility, whether we are living in the right lifestyle or not, depending on our, what kind of genetic cards we've been dealt, we have to understand how that genetic information is actually decoded. And so, you'll be hearing a lot of words such as genomics itself. What does that actually mean? That really means trying to interpret the 3 billion base pairs of DNA in a way that we can understand how each individual's genetic output will affect their lives. And that's an extremely difficult challenge to do, because the 98% of the genome sequences that we don't know how to read is regulating that whole process. So it's the regulatory elements of that genome that are a big challenge, and we really need the help of our bioinformatics friends and our biophysicists to try to understand and work out the mechanisms that control gene expression. You also have heard the word proteomics. So this is the other part. Even that 2% of the genome that we can read, which is the coding part, that's where all of the proteins that run all the metabolic processes of a living cell are encoded in. Nevertheless, of the 20 to 30,000 genes there, which is only 2% of the genome, very small percentage of those genes products, in other words, what they actually do, what the functions of these genes is not known. And that's why every year we find more genes that are involved in cancer or more genes that are involved in diabetes, because in fact we've really, even the part that we can read, we've not understood it entirely. And then finally, we run into a really interesting challenge which brings this audience particularly into mind, and that is that we've crossed over from those of us working on the genetics and the genomes of humans and organisms, trying to read the genome, to trying to interpret that information for you. That is, how are you going to make use of this information? So I had a very interesting conversation with Jim Watson about this after he got part of his genome done. And he came to me and he said, Tej, I've been really curious about this. And he says, I'm really glad I didn't see my genome until I was 80. Because if I had, they would have locked me in a dark room because I was susceptible to this disease and that disease, and it would have had a huge impact on my life. So his usual sort of quip was, nobody should get their genome done until they're 80. So obviously we have a problem that even when we do learn how to read the genome information, what are we going to do with that information? Now, there is a bit of a silver lining here. I think that already, even with the small amount of information we have, we can already make some predictions about drug susceptibility or side effects of certain treatments and lifestyle. And one of the most exciting to me is a little known area where, because of the large differences in polymorphisms, what we call the polymorphisms, what you just heard is the 1 or 2% differences between individuals, that might actually give us a key about some really simple things, like some people might want to take this vitamin more than that vitamin. In other words, micronutrients Not big drugs, but micronutrients that could have a big impact on your life depending on what kind of genetic cards you've been dealt. So I do see this information just growing rapidly. So I'm very optimistic about it, but just keep in mind that we're at the very beginning of this road.
Okay, so here we are in the genomic era. What does that imply for medical science and medical care? In 5 minutes or less, in what I hope is accessible language. Well, it opens up a number of prospects. It opens up new ways to examine the cause of disease, and I now extend that to many diseases as opposed to cancer, which we talked about in the previous session. Our individual susceptibilities to disease. Not everyone who smokes gets lung cancer. Not everyone who gets infected with poliovirus gets a paralytic disease, not by far. Why is that? It provides new approaches to targets for therapy. That was dramatized for you in the cancer session. It allows us, it will, we hope, allow us to predict individual responses to therapy, both the beneficial responses and the noxious side effects. And it tempts us to contemplate re-engineering the human genome to what we might think would be improvement of human welfare. I'm going to comment on several of these. But first, I want to just make a few remarks about ascertainment. What data do we need to pursue these seeming advantages? Well, Bob Tijan alluded to the fact that we actually haven't read all the information in the genome. So a lot of what we do is deal with anonymous variations among our genomes. And there are about 7 million presently available that are useful in such surveys. So using high throughput, and I am covering a lot of sophisticated territory here, using high throughput techniques, we can survey of large populations, ascertain which, if any, of these variations among us seem to be linked to one or another disease, one or another human quality. Even risk-taking has been pointed to on several positions in our genome, particularly in Craig Venner's genome, incidentally, which was spread across a full page of the Science Times section about 3 weeks ago, including a locus for sticky brown earwax. All right, so that's what we do, and it requires immense statistical power beyond anything that has ever been employed in biology before. It involves new technologies which need to get faster and less expensive, and TIJ, as we call them, alluded to that. And again, we are dealing with biomarkers, not necessarily identified genes, but the markers tell us where to look for responsible genes. Now, there are two sorts of genes, strong determinants, or two sorts of determinants, strong and weak. Strong would be hemophilia. Inherit the bad hemophilia gene, you get the disease. With a stretch, the BRCA genes of breast cancer. Inherit a bad version of that, one of those genes, and you have a very high predisposition to breast or ovarian cancer. But then there are the weak determinants, which are going to figure much larger in what we're talking about. In and of themselves, each of these probably does not signify a huge risk, but they somehow combine to create a risk, and it is these that are going to play the largest role in doing much of the prediction that I alluded to at the beginning. We have a rapidly expanding database. Within the past year, and I only kept a short list because this is not my immediate field, weak determinants of susceptibility have been identified for acute lymphocytic leukemia. You heard about that before. This is the most important pediatric cancer. Breast cancer, tendency to obesity, type 2 diabetes, cardiovascular disease, and multiple sclerosis, just within the past few years. In the case of cardiovascular disease, there were 9 separate positions on the genome that were somehow implicated in a predisposition to cardiovascular disease. Each one of those is weak. Put them together, and you get a measurable predisposition. Now, there are some complications in all this. In the case of the strong determinants, there are daunting personal decisions. Think of Huntington's disease. A medical geneticist can tell you if you're in a Huntington's family whether you've got the bad gene or not, but there's nothing to be done about it. When, if ever, do you want to know that before the disease begins? BRCA gene, just within the past week or two, there was a riveting account in the New York Times about a 35-year-old woman who was training in obstetrics and gynecology, and you would have thought that she would